Welcome everyone! So today in this video, we'll be talking about DNA replication and protein synthesis. But first we start with the deoxyribonucleic acid or DNA. DNA plays a big role in our body as it contains the genetic material that contains all genetic traits, thus called genes. But where is this located? Now there's two kinds of cells, eukaryotic and prokaryotic cells. In eukaryotic, DNA is contained in the nucleus, but in prokaryotic cells, DNA is in the cytoplasm because they don't have a nucleus. Sad to be them. Now let me introduce to you nucleotide, which is the building block of DNA, and it contains the following parts: the 4 bases, the 5 carbon deoxyribose, and the phosphate group. The bases are adenine partnered with thymine and cytosine partnered with guanine. These are the 4 bases that is linked together by a hydrogen bond to form DNA. Now that you know the basic information about DNA, let's talk about DNA replication, which is the process of replicating DNA. The process starts with your DNA strand being unwinded and flattened for our main DNA replication characters to work their way in replicating DNA. Let's meet the characters. First, we have helicase, which unzips the 2 strands of DNA, separating them from each other. From those 2 strands form the so-called leading strand and the lagging strand. The leading strand, from the word lead, replicates in a continuous process. It first starts from the primase, which puts the starting line from which the DNA polymerase can work their way through. DNA polymerase is another key character in the process of DNA replication. It is also called as the builder. Here comes the DNA polymerase, which will be replicating a new strand from the old strand of DNA. It goes from 3' to 5'. Now let's go on to the lagging strand. From the word lagging, the process of replicating DNA lags as the strand goes from 5' to 3'. It forms these so-called Okazaki fragments, which are small portions of DNA strands made by the DNA polymerase. After replicating these DNA strands, the enzyme ligase comes in the process and seals up these strands. Disclaimer: what we're teaching and showing is only a small portion of the big picture. There are other players in the replicating process of DNA, but we're only mentioning the main ones.
Protein synthesis. Here comes the RNA polymerase, which will be making the strands that will serve as a basis for protein synthesis. This process is called transcription. The mRNA or messenger RNA will then go outside the nucleus into the ribosomes to create proteins. There are also main key players in the process of protein synthesis, namely the messenger RNA, ribosomal RNA, and transfer RNA. The rRNA is called the protein builders of the cell and are responsible of reading and the order of amino acids and linking amino acids together. The tRNA or transfer RNA is one which transfers 3 anticodons that are complementary to template made, which makes an amino acid. As the 3 complementary pairs are called codons pairs to each other, it releases the amino acid and binds to another amino acid, which creates this bond called peptide bond, which then turns into proteins. Thank you for watching, and we hope you have learned something.